Thank you. All right. So let me just put our notes document in the chat. So please fill out we're collecting a list of participants. So if you could fill out your name and institution in that notes document, that would be great for our records. I've also put a short agenda there and some discussion questions that I'd like us to talk about a little later. But I thought first today I'd give a presentation on the work that we've been doing as part of BBOP and as part of this Omics eDNA task team with the OBPS. So I can share my screen. And does anyone have any questions? Everyone is able to access the notes document? Looks good. Okay. Let's see. All right, does that, everyone's seeing presenter mode, not the, yeah.
Yeah,
great.
looks good.
Okay, so thanks everyone for joining our session. So we've been working since 2020 as part of a groups in the Omics and eDNA task force within the OBPS, thinking about how we can work towards better sharing protocols within Omics and eDNA. And so today I'm going to talk to you about kind of, the work that we've been doing since then. And this has evolved into a project with the UN Ocean Decade called the Better Biomolecular Ocean Practices Project, or BBOP. But first I thought I'd introduce real quick OBON, because we're a project that's under this larger UN Ocean Decade program. And so OBON, which stands for Ocean Biomolecular Observing Network, is this large program as part of the UN Ocean Decade. And I've put their objectives here, but they want to help us, better understand ocean life by analyzing biomolecules. So really the goal is to build this coastal to open ocean multi-omics biodiversity observing system. So that's the large vision. And then we'd like to develop and transfer capacity to help to have additional observation activities around the world. And then also use this omics and biomolecular information to enhance marine ecosystem models and also address scientific management and policy questions. But this is the larger program that we applied to be a project under with BBOP. So this is our larger framework. And so what was the motivation behind all the work we're doing? A lot of us work in observing biodiversity through omics or eDNA techniques. And so we saw this really clear need in 2020 at the first OBPS workshop that I attended, where we need to share detailed protocols across our entire value chain in an omics study, from designing the experiment, collecting the samples, analyzing the data, and then sharing that data. And so as a group, we came together and started talking about how we can make this system better. And we were working within the OBPS. And so we formed this task team for omics and eDNA protocol management. And so this, what we worked towards was thinking about ways that we could create a better way of sharing these protocols. And so this kind of led into this BBOP project. And so, we designed a framework of mapping omic protocols and data workflows. So here on the right is a figure from a paper that we published where we mapped out a metabarcoding study. And then our vision was that we could take each chunk of protocols and map out an entire study from start to finish. And so I'm actually just gonna flip ahead here just because I want to show we developed metadata for each omics protocol. So each of those chunks, we define different terms that describe those protocols. So we have which type of protocol it is, for example. Is it a sampling collection protocol for how you'd sample eDNA? And then terms about the project, the purpose of the protocol, what resources you might need, the type of analyses used in the protocol, and then terms about where that protocol has been applied, such as geographic location, environmental context, and what type of material you're sampling and what's your target taxa. And so we thought if we have these metadata terms to describe the protocols, it would help us find them and organize them better to be able to better discover protocols. And so as part of this paper, we also define these different categories of kinds of protocols. So here is again this metabarcoding study workflow. And we've defined different kind of broad categories that each of these protocols can fit into. And we really saw the utility of this being that, Omics and eDNA, systems of protocols are kind of complex, going from experimental design through final release of the data. And so by splitting up these large protocols into different chunks, well, we can better see where similarities are occurring across different research projects. So say someone might be using the same DNA extraction protocol as someone else, but a different PCR amplification protocol. And so we thought by separating out these different kind of chunks of protocols, we could better have a system where we could compare workflows between groups. And so as part of our work, we've moved to creating a GitHub repository. So these metadata terms that describe our protocols, or we call them MIOP, minimum information about a OMIC protocol. So we've been developing those terms as well as these as protocol templates, which we've made in a markdown template format. So the benefit of making these protocol templates and putting them on GitHub is that we can track changes across protocols. So say in a PCR protocol, you have a change in your annealing temperature or a change in your primer that you're using. It'll be easy to see exactly where that change is occurring in your protocol and then compare it to past versions. And that's really what GitHub does for us, is it like software, you can see how a protocol is changing through time. And then it allows us to also create a unique DOI for each version of that protocol that we could link to the data. And also a nice thing about GitHub is it has free and open access, and many others who are already working in the bioinformatics sphere are familiar with GitHub too. So these templates that we've created, we have in a separate repository, and we've created a system of templates. And our vision is that you can take these templates that we've created and then fill them in for your specific institution. So the advantage of these templates too is they're machine readable. So then we could start to compare between institutions what people are doing. And just an example, markdown format, it can be a learning hurdle if you haven't used it before. But what's nice about it is this machine readability and the ability to track changes, but also that you can export a PDF at the end. So here, for example, is one of our PCR templates that you would fill out with your specific information. But at the end, you'd end up with a format like this, where you'd have common tables. So somebody who's looking at your protocol could interpret it easily. So our vision is that, through a typical, for example, metabarcoding study, say you wanted to start your own study, you could choose what type of protocol you needed, and then use these different metadata terms about the different protocols to compare them. So what we wanted to do is develop a framework, almost moving towards this decision tree concept, but a way of comparing different protocols to each other very easily. And so, for our sessions this week, we thought first today, we'd really focus in on talking with others who are also working on different efforts to harmonize OMIC protocols and, go over what we've done so far. And then tomorrow, really focus on kind of those protocol metadata terms that we've developed so far. And then finally, our third day will be led by Inés, and there we'll be really talking about a lot of the biological and physical challenges in the field of sampling eDNA. So what goes into making a good eDNA field sampling study and discussions around that. So I thought first I'd also share today what I thought some of our good outputs could be, but we'd really like to, better survey who else is also working in this sphere. and if there's potential points of convergence, so where we can work together, and then get feedback on the protocol templates that we developed so far. But I'm going to start out with first 3 questions, and maybe I'll stop sharing here because these are actually in our notes document too. But let's see, it looks like there's 31 of us. And I thought maybe we could go around and give a quick introduction, and then say if you're part of another initiative or effort working towards method harmonization. And if so, maybe what are your big objectives there? Or what is your primary goal as part of that objective? Let's see. And I can call out to people. All right, so I'm going to just, maybe I could have my co-conveners also introduce themselves real quick. Christina, would you like to introduce yourself?
Yeah, sure. I'm Christina Pavloudi. I'm a postdoc researcher currently at the George Washington University in the United States. I have been involved with a task group for the eDNA and metabarcoding since its beginning. And I was involved in my previous institute in HCMR in Greece. I was involved in initiatives such as the ARMS MBON or the IMOBON. And there was a lot of protocol sharing, protocol description, and so on and so forth. So yeah, I'm not currently in any other initiative that aims to harmonize data sharing and protocol sharing. So yeah, that's from me.
Thank you. Raisa, would you like to introduce yourself?
Yeah, sure. Thanks, Katie, and thanks for giving a great introduction to the work that we've been doing so far. So my name is Raisa Maya. I'm currently doing my PhD at the Alfred Wegener Institute in Germany. And I'm with Robin Samuel, who's unfortunately not able to join us today, Katie, Neil Davis, and then also Chris Meyer. Well, Neil and Katie, Robin and I were the one who kind of started the task group with the first session that we convened at the OBPS workshop, I think 2 or 3 years ago.
It was 2020, yeah.
Yeah, exactly. So quite some time ago, and that's where we also met all of the participants, or many of the participants that we have in the task group now. So Katie already mentioned the OBON program that BBOP is under, and I'm part of that as one of the ISAC members, because a lot of the work that I'm doing is about harmonizing or building interoperable networks of data exchange between omics observatories. So within that, we have OBON, and now we also have OmicBON, an Omic Biodiversity Observation Network under GeoBON. So really making sure that we not only look at the marine side, but also then connecting that through GeoBON to the terrestrial atmospheric observation using omics techniques. And the methods that we're using are such an essential part of that. I've been involved through Pierre and some others, Pierluigi Buttigieg, who's also on the call, in the discussions with the data coordination group under the UN decade. So also a lot of that, or some of the strategic challenges that we identified there, were really about how can we trust the data and how can we develop capacity around what we're doing here. And that's really where I can see that straight connection to what BBOP and the OBPS task team are doing in making sure that we know how any data was produced, and that we can understand that a lot more easily by making it machine readable, having the metadata that very clearly already gives you an idea of how the data was produced. So yeah, really glad to be here, glad to have another session with everyone and get some input from outside of the task group and the project. And yeah, I'm looking forward to a great session. Thanks, Katie.
Thanks, Raisa. So let me just put, I thought the first kind of introductory questions. We could go around and please introduce yourself. And then if you happen to be part of another initiative, if you could briefly share, what are the main objectives and what are you working towards a method for better sharing protocols? Anais, would you like to introduce yourself too? Sorry, Anais will be leading especially the third session this week.
Yeah, I can start. Or I can introduce myself. So I'm Anais Lacourcière. I'm in Canada. Lived in Canada in New Brunswick. So I work for the government of Canada, Fisheries and Ocean Canada as a researcher. Since 5 years, I work on eDNA since 10 years. Sorry if you hear the kids, it's 5 years, so the kids are running around. So basically our role as being a research scientist for the government of Canada is making sure that we innovate tools for the monitoring the coastal environment and providing advice on how to interpret data and making sure that we're having new tools and being innovative. Our second role is to make sure that the communication that can be used properly by managers that need to take decisions. So making sure that we are flagging red flags and those type of things. So in that role, basically, I've been part of what we call CSAS. It's the Government of Canada report, where we basically they ask us first, the managers, how to standardizing. So they want to standardizing eDNA. I see the question a lot more complex than the basic standardization. So basically, what we decided to do is to start by standardization of how data will be reported, including metadata, instead of providing a standard report. The standard, so it was mainly, okay, you can do whatever you want, but make sure you report it and all of the details. And as a manager, if you don't have those information, I will not be able to inform you if the data are properly done and all of the steps have been well considered. So basically, it's so that's been a big step for us. So now managers can use eDNA. One of the research project, and I just got funding a couple months ago, but that's for the next couple of years. I'm doing a meta-analysis on optimizing the detection period. So basically making sure that we have guidelines on when to survey. So for that, we do a meta-analysis, including trying to understand, okay, you have all of those papers that have been published. How can we learn now? Like we all know that we need to consider reproduction period, physical factors, and those type of things, but how can we really, if someone is starting, we know that eDNA is used by not just expert scientists, but it's used also by people that, what we call here eDNA users. So they really need to learn about population and community and not all of the information around. So that project really trying to just compile all of this data, including the methods. That started, I was like, okay, we need to make sure that all the protocols are well entered within the template we have. And how do we visualizing it at? How so that will be on public, we're developing a public online tool, basically, where people can go try to see and having a link. So for me, it's the things that I'm hoping to learn is really, is there similar initiative where people get those type of information when they design their survey? If you start a survey, the people will always want to go in the field and having a lot of fundings. And the first question is when to go, in my perspective. And after that, there's all of the labs. So most of the literature review, in my perspective, has been mainly on lab protocols, whereas for me, my role is more about sampling design and how to interpret the time series data in the long term. So if we see changes between the year, how can you compare multi-year? And how managers can be sure if they don't detecting it, what all of the factors that can create it, not just the fact that you can consider the species is not there, even if you don't detect it. So we need to make sure that the proper management decision can be taken. So that's why I'm here, to learn what's the similar initiative and how do you do that?
Thanks so much, Anais. Jody, would you like to go next and introduce yourself?
Thanks, Katie. Good morning, everyone. I'm Jodie Vanderkamp from CSIRO in Hobart. I'm part of the Australian Microbiome Initiative, and we are trying to provide microbiome data from all across Australia through to the coast and the open ocean. We have two core data sets which are completely standardised, and in the terrestrial space, that's through National Parks Australia. And then in the marine space, I lead the IMOS, Integrated Marine Observing System Marine Microbiome Facility, and we provide standardised data from the national reference stations as well as some coastal sites. What we realised pretty early in the piece, though, even though those core data sets are standardised, not everything can be that we want to bring into the database. You know, not everyone has the same equipment, the different systems you're working in. And so what became really important to us, and why I wanted to be part of this task force and think it's a really important piece of work, is recording metadata about those samples. So everyone that contributes data to the Australian microbiome has to provide really detailed protocols. That all gets recorded in our scientific manual, and each protocol gets given a unique number. And so then when people download microbiome data, if they want to try and do a regional analysis, so bringing lots of different projects together, or even as part of a global analysis, that metadata that comes down includes that information about how the sample was produced. So it'll have sample collection by method 1.2, you know, DNA extraction method 2.0, or, you know, chemistry via this other method. And so I think, yeah, what we're doing in this space is really important, and it'll be really great to get other people's feedback.
Thanks, Jodie. And I guess I want to mention too, in our notes document with, which I can share the link for again, in case somebody has come in late. We'd like to kind of create a list of all the different initiatives working, that people who are here, what initiatives they may be part of working towards Omics protocol harmonization. And so if you want to go ahead as we're going through these introductions and just start, just place yours within that list, I think that would be great too. So we can start building that list. But Sam Lake, would you like to go next? I'm just going to work through my screen.
Sure. So I have been here very much as an observer. I'm not involved in Omics, but I've been involved in some larger overreaching organisations such as the Marginalised Zone Cruise, which the Australian Antarctic Division and the Australian Antarctic Partnership are organising for next year. And also SOOS, the Southern Ocean Observation System, and which is a coordinating body for Southern Ocean science. And yeah, last year I did a contract with the UN, writing best practice manual and a comparison of data variables and data codes for sea ice. So I've had an introduction to the topic, and I'm interested to see how all the different data types, oceanic data types, are organising themselves. Thank you.
Thank you. Noria, I'm sorry, I might butcher some people's names. But Noria Mohammed Salah, would you like to go next? Oh, sorry. Would someone like to, maybe it would be better if someone just wants to volunteer and jump in too, or if people are part of initiatives and they want to share real quick what they're part of.
I can go.
Thank you.
All right, it seems my microphone is working again. Good morning, everyone. It's pretty early for me. My name is Martin. I'm in Australia. I'm with CSIRO, so the same organization as Jodie. So I'm a postdoc here, and what I'm working on is helping integration of eDNA methods in marine park monitoring in Australia. As part of that work, we've just, with a group of eDNA researchers in Australia, produced best practice guidelines for eDNA in monitoring. So we've done 2 documents. The first document is general, is basically, it's not as detailed as what's been discussed already today. The first document is guidelines on how to really build your own SOPs. The reason for this, the guidelines we've done are not ocean specific, so they're general eDNA guidelines. They were at request of the government of Australia, so I'm not sure how to, like the Department for Agriculture, essentially, agriculture, biosecurity, all of those things. So that originally came from the biosecurity area, where they asked colleagues of mine to organize some sort of standard, and in quite a large collaborative effort with, I think the first workshop, we had about 60 researchers from Australia. We came to the conclusion you can't have one SOP or one standard for all eDNA work. So we've written instead a guideline document on how to really develop them, or how to run an eDNA project, depending whether it's biosecurity terrestrial or whether it's marine monitoring. So it's quite a broad document. I'll share the link in the chat in a second. The other document is a standard validation guidelines. So again, relatively general. There are movements in other members of that document who've been involved on creating more specific SOPs and standards. So those will come out in the future. Another thing that is probably worth mentioning here, I'm also part of the very newly minted Southern eDNA Society, which is going to be the scientific society for eDNA in Australia and New Zealand. So at the moment, we're at the stage of the formalization of the society. So all the legal paperwork, essentially. So in that group, I lead the standardization work, standardization group. We've got a communication group, but the details of the society still need to be worked out. They will be, we'll officially announce everything on the eDNA conference, the Australia-New Zealand eDNA conference that's happening next, I need to think, March? No, it's February, sorry, mid-February. We had a COVID postponement, so things change a bit. So yeah, that's what's happening here in Australia. So in short, a new society and best practice guidelines, or a way towards best practice guidelines.
Thanks, that sounds great. I think we agree that it's hard to agree on an exact protocol to use. So it's instead important to talk about the best way to develop those protocols or share those protocols. Would anyone else like to jump in?
I can go next. Can you hear me? Hello.
We can.
So I'm Julia Santi. I'm working for the European Marine Omics Biodiversity Observation Network. That's an initiative from the BRC. And yes, we are working on, I'm working on an initiative on method harmonization. And in fact, we have produced a sampling protocol for omics for different habitats, and we have collected all those in one large document that's public. It's shared, it's on the OBPS, and we basically use those protocols for our 19 observatories, trying to have everyone collect comparable samples and then continue with the analysis. We are now working on the next steps, let's say, protocols. We are discussing metaport coding and metagenomic protocols a lot in our community, but we haven't progressed yet. However, we do plan to have an updated version next year because this is an ongoing process. We are discussing the problems we are having, trying to solve them, trying to make specific, let's say, modules for specific regions. For example, when you have severe blooming periods and you cannot filter seawater, and we want to update on some of those details. And yes, we are sharing the protocols. They are open for everyone. They are on OBPS and on our websites. And we haven't worked on how to share them further than that. However, we have a few ideas we are now discussing with our Open Science group. And those ideas include, for example, creating keywords for the protocols, identify what the bits that are more important, let's say, for comparing protocols at a later stage. And this is very compatible with what you described. And I've seen other discussions from this group also in the open presentations. So we really want to engage and discuss this further. So yeah, I'm also looking forward for this session and today and the next and the day after, and to engaging with all of you even more.
Thank you so much. That would be, I should also say that this group meets about every 2 weeks to try to further our, definitions of MyOp and our protocol templates, and anyone is welcome to join, and we'd love to have, others join us and others try to, put their protocols into this format too. We're at the stage where we're trying to test how well it works for different groups as well. Would anyone else like to jump in and share?
I can go. Happy to. No, go ahead.
Yeah. Hi, I'm Jonathan. I'm based in Brazil. I'm partially working with the Freshwater Bond group, looking at standards there and how eDNA can be used. I've also got a lot of projects here and trying to develop a Latin American eDNA network. So I try to apply the standards that we're developing in a more general background here. Yeah, I guess that's it. Very simple, very quick.
Thanks. Frank, would you like to jump in?
Yeah, thank you, Katie. I think that this is a very important effort. I'm Frank Miller-Carger at the University of South Florida in the United States, in St. Petersburg. I'm an oceanographer, and I'm involved in a number of these initiatives that are trying to bring these different groups together to talk about these protocols and how we make them, how do you harmonize them? One of them is the Marine Biodiversity Observation Network, or MBON, which is also linked through GeoBond, through the Omic bond that Raisa mentioned. And in the Ocean Decade, we're leading something called Marine Life 2030, which is also linked to OBON and several other marine life related programs. So my interest in all this is a concern and an interest. Of course, we want to see the application of eDNA in a practical manner. Know what kind of problems, societally relevant problems, can you solve by using these methods, and to enhance any other type of monitoring that we have. And so that's the concern when you have so many protocols and so many different groups developing their own metadata and protocols. How do we get convergence? Now, I think this group is really, really key in trying to foster that dialogue on convergence. And then having case studies on how do you apply this beyond the research that we all do from an academic point of view. How do you actually apply this to solve a problem? So I'd like to hear actual examples, and I'm very encouraged to see what IMOS is doing and really bringing this into an operational setting. So that's a good example for all of us. Thanks.
Thank you. Would anyone else like to jump in? Oh, Jay, I think you're muted.
Yeah, it works better when I don't mute. I'm interested in the propagation of best practices at a global level. As co-chair of the Ocean Best Practice System and one of the organizers of the conference here, I think the task team from the last 2 years has really been exciting and very important. I do agree with Frank that if some of the work that Jody's doing can be expanded, it'd be worthwhile. make only one additional comment. We go through convergence as a process, but there's also a process of endorsement where experts get together and they look at all the practices which have identical aims and try and sort out which ones to recommend to newcomers, people who see these myriad of practices and don't know which one to choose. So we might consider endorsement as part of the discussion sometime in the next few days. Thank you.
Thanks, Jay. Yeah, I think that's an important question of how to, for people who are just getting started, how can they tell what they should, or what would be like the best practice implement in their region? And one thing we had talked about is some of the reasons why we have these different metadata terms of what geography the protocol has been applied in, or even we discussed listing out what equipment might be necessary in a protocol, just so that people know what's been applied to the same kind of region they want to work in. And then also depending on their needs or their available resources, what protocols might be suitable for them. But that's definitely a discussion we want to have, I think, in the next couple of days.
Yeah, just to jump in and comment, if we had a similar discussion also last year in the previous OBPS workshop, we were thinking of when we were discussing a lot about the MIOP and what other fields there should be included, and people were mentioning something like an endorsement, a protocol endorsement, like method, like a star system, so that some things would pop up, like the most popular ones and things. Yeah, there were some good ideas last year. Yeah, maybe we can think about it more this year.
Mm-hmm.
So also just to mention, one of our goals with BBOP is working with the projects that are working with, these larger programs to just show what protocols they're using right now. And so I think it's like a hard thing in our field to know who's doing what and where. And so I think part of our project is just trying to increase the visibility. So people who want to maybe get into this field too can see, okay, what are these projects that are working with OBON? What kinds of methods are they using right now? And so I think that can be really helpful as well. And so we also welcome, please, we're looking for others who would like to share their protocols through our system too. And so if you're interested, you can contact me or Reza, and we'd like to work with you to, put your protocols into the system on GitHub as well. But would anyone else like to share?
Yeah, I would like to see.
Yeah, that's okay. You go ahead. I was just going to comment about the endorsement part.
Oh, go ahead. I will. For me, it's
just very quick. So I'm sorry, I'm not sharing my video. I'm a bit sick at home and I don't look very good. But I'm from IMOS and we do endorse the best practices from our facilities in Australia. And given we will be endorsing Georgie's work, especially because it is clearly passing through the global experts, which is your group. So you have global experts from different places in the different countries and different protocols, and she is taking into account all of that. And so that is one of the big tick boxes right there. And yeah, so like Jodie's work will definitely be endorsed by IMOS, and then if your group ends up having a best practice for all of you, like that will end up being endorsed by us indirectly. So just wanted to clarify that. I don't know if that's an important thing to say, but I felt that was part of the conversation. So sorry, excuse me.
Oh, thank you. And thanks for joining even when you're sick.
No, that's okay.
Well, one of my question, I guess the group is how in my lab we do use 3 different protocols, and they are all really good for a specific environment. So basically, there's no one best practices. There's one best type of filters for when the water is turbid, whereas you have a different type of filters if the water is very clear, coastal environment, and freshwater will be a different one. And then, of course, it changes the filtration type you're using, and then it changes the extraction because it's not the same type of filters, so you're changing all of that. So that's where basically I found very difficult to find the best practices. And this is the same things for when invasive species. They are one of our main clients, the managers working for invasive species. And can I use the same time, the same sampling period, which is optimal in Europe for the same species than in Canada? So that's really where I found that very difficult, is to have information on how can we get there? How can we have really guidance on which protocols using in which environment, and and I found it very difficult. I see a hands up from Frank. Any comments or recommendation?
Yeah, nice. And I don't know that there's an easy solution to that, because life is so varied and crazy, and every application that we can think of may require a specific method for a specific problem. And yet, I think somebody else said before, you want to compare things over time and from one place to another. And one of the big questions that we all have, and that is so present right now in international discussions, is biodiversity changing? So how do we get at that? And so that's the hope, is that eDNA and these omics techniques can help address that. And yet, if we don't come to a minimum set of variables that we can all measure the same way, like people measure temperature or you measure wind, there's certain methods, units, you know what you're measuring. You hope at least that the instruments are calibrated the same way, and then you can compare temperature from one place to another and say, okay, we have climate change. So they can talk about change in a way that makes sense to a lot of people. So, and that's one measurement. And so I think that the people that look at climate look at maybe 7 variables. We look at millions of variables, and we don't really know how to talk about change, except at a very localized place. So I wonder if there's a way to think of, can there be an agreement around the world on a minimum set of things that we can measure in a particular way, so that we can compare some metrics of biodiversity using omics? Or is that a rhetorical question, or is that an impossibility, or how do we address this?
Martin, you raised your hand. I don't know if you want to comment to that or comment.
Yeah, I do actually. I completely agree with you, Frank. It's a very good point. The thing is, that's a problem that's not unique to omics. Maybe it's different in biochemistry and those things, but when I look at my personal work with marine parks, what is usually used in methods here to measure those changes. So that's what managers are after. They're not necessarily after specific metrics, but they want to see whether their decisions or whether their actions create a change. Like, do you create a protected area? Does it change? What changes is what's measured kind of doesn't, I mean, it does matter, but it's less relevant than knowing whether or not it goes up or down, basically. So it's not unique to omics, because what is currently used across Australia, we've got such a big marine park area, right? Is a wide range of methods that are visual. And some people will use beta remote underwater videos, and others will use diver operator videos, and others will use underwater visual surveys and ROVs. And there's such a wide range of methods. And even, for example, one of the most common used ones, the baited remote videos here in Australia on the east and the west coast, they've got slightly different protocols. So they have the same problem, and people get on with it. Management gets on with it because they have within their park the same metrics have been used for a certain time. And so for some of those methods, like the BRUVs, the baited remote underwater videos, they're starting to get through different programs in Australia, which Rachel explained last week. So they're starting to get some standardization, but even then, that's still not there. And that's a method that's been actively used in monitoring for, I think, about 20, 30 years in Australia. So it takes a while. And so because we've got such a wide range of different, as you explained, right, of different methods, it'll take a while. And I think what matters really is that metadata, because that's what the lessons I've learned from the BRUV standardization people is, it's all the metadata that counts to, in the end, bring it all together. So like, yeah, we ideally what you suggest would be the perfect situation, but I don't see that happening anytime soon. So the initiatives like that have been discussed today, the metadata would be the key to bring it together afterwards. What is really important though is that within the same region or same management unit, that there you use at least a comparable method. But yeah, within the visual surveys, especially globally, there's so much variation that, and people don't, I mean, it's kind of acknowledged. It's a very, because I come from a non-genomics background, and in visual surveys, it's very often glanced over. They go, someone else observed the fishes in the same region. Oh, we'll just compare it anyway, even though there will be a great variation. So it's, I think as a genomics community, we're a lot more aware of those differences in protocols, and a lot more hung up is not the right word, but we care a lot more than say the visual surveys, which I think is a good thing. It is a good thing, but we can't, like, I think we should be realistic, really. But it would be brilliant, your idea. It would be fantastic, but how to do it, I don't really know. I wouldn't even know for Australia, let alone for the whole world.
So I guess for today's discussion, I had a couple of questions that were a bit more detailed, but we're almost skipping to kind of talking about the last one that I had for today is, you know, what do we think are the main challenges surrounding protocol sharing or harmonization omics? And do we see points of convergence across efforts? And so I think, you know, we've already brought up some good points that are like points that I just agree with, that, you know, it's hard to find, you know, a best method because best depends on where you're working. And so it's really about making sure when we look at data, we know exactly how that data was generated, and try to create these infrastructures for sharing protocols better. But I don't know if I can put that question kind of in the chat, but I don't know if anybody else wants to chime in about, you know, what they think.
Well, going back to Martin's, Katie, and this question on comparing different protocols, I guess all the protocols must be findable and comparable. I mean, if some group has a protocol or a metadata format, but that is not widely known, so how do you find the different metadata formats and then find the different metadata that people are putting out there? Because at the moment, that's another thing. Is that I'm thinking that you're probably going, I'm not sure exactly where you're going to put the metadata yourself, but I think you're talking about OBPS as one instance, but there may be other places where the metadata gets stored, and then you have to find it among the different omics or whatever other method protocols to compare them. And that's another complication.
Right. So the metadata we've been developing just to describe protocols, we're thinking of attaching to the protocol itself, and to help be a way of sorting and seeing where protocols have been applied. So describing the methods. But then, you know, we're also talking about all the metadata that gets attached to the data itself.
I just thought I'd sorry, Raisa. Just sort of around those challenges, you know, protocols are only as good as the detail that's put into them. And, you know, even though we are trying really hard to get protocols into the Australian microbiome and attaching that to the data, you know, quite often what I think is enough detail in a protocol of mine, when I share that with someone else, they read it and they're like, oh, I don't understand what that bit, how you move to the next step, or how did you do that? And that's one of the reasons I think these templates are really important. But at the same time, we have to get buy-in from the community. So they're only going to be good if we can get people to actually fill them in and fill them in well. You know, we're all time poor. So I think that's going to be a challenge for us. But if we can get a critical mass where there's a lot of us already doing it, then I think we'll sort of get that tidal wave of, oh, we better get on board, fear of missing out, all of that sort of thing.
Yeah, that's actually similar to what I was thinking about, that we really need, everybody is so busy and everybody has so much work to do. So there's not, at least currently, there's not really any way to incentivize having a really good protocol that you can share with anyone around the world and anyone can read it and understand it, not only humans, but also machines. So if we can, yeah, I feel like that will be one of the critical steps in actually incentivizing the extra hours put in. We've already, like with developing the templates, like you can get a student assistant, get your, like take the protocol that you have, write it in that format. It doesn't take long, but it still takes long enough to provide a hurdle to a lot of people. So I think, yeah, in the context of OBON, so the program that BBOP is under, I think with that, we really have an opportunity to get some really important projects and their protocols into the formats and to test the template, to test the metadata. And with that, then make sure that we have at least part of that critical mass already that you mentioned, Jodie. And it's really great to have you from the Australian side, from the Australian microbiome in that as well, to make sure that we get your protocols in there. So I think with us coming together as people and with us coming together under OBON, we really have a chance to get the critical mass ready, and then hopefully we will have a way of finding some other ways to incentivize it further.
Thanks, Raisa. Anais, would you like to add to that?
Yeah, I think it's a very important question as well this time. And basically with eDNA, what we found is that most of the projects are done by many different people. And basically, at least in our government, it's mainly at the end about the researcher that needs to fill, you know, the data and making the link with the international database and things. Whereas all of this step has been done by sometimes completely different private company, provincial government, then you have the labs, and then you have like the people that have sent samples and things like that. And in my perspective, one of the best way it's make it easy, very easy accessible. Basically, it's probably app on iPad or something like that, where everybody has a project ID, and then just when you're in the field, you just that project ID, and then everything is linked together, and not like at the final step. So that's how I see that feasible. If not, we're trying now, and each lab are providing different protocols and different and its report, and it's very, very have full-time person working on just a couple projects to compile together. So make it easy as an app. I think that's gold.
One thing that I really like about the GitHub system that we're trying to develop, or that we're developed, is that, you know, in our lab, we're often changing, we have a protocol that we've been using, and then we want to change one detail or one part of it. And so it's often these kind of minor changes, which are very important to record which data sets are affected by that change, but just keeping track of when that change occurred and which sequence plates it applies to, I think can be very difficult. And so having a system where you can have like a separate, you know, a timestamp and a separate DOI for when that change occurred and link it to your data sets is very important for our lab. And so I kind of like the modular system that we developed for that reason, because it's not often that we're changing everything in our pipeline. We're often making small changes to one part, or maybe we're altering our, you know, our DNA extraction protocol slightly. So it's really about keeping track of all those small changes, which we know we have to keep track of. It can be a difficult task.
Yeah, and also just to comment, the good thing with GitHub is that nothing is actually lost. There's like a history of all the changes. So even if you change something in your protocol, or you create a new protocol based on another one and you change a few things, you can always go back and see what you have changed and what was the original version and so on and so forth, which makes it really easy and looking into the future really, really helpful, at least as a, into my thinking. Yeah.
So one other discussion point that I wanted to talk about today. So a lot of times we kind of think a lot about our lab processes, or, trying to compare between different PCR protocols or different sequencing protocols. I was wondering if generally today, which portion of the OMIC workflow do you guys think about most? Is it, sample design or sampling procedure itself, or do you also think about protocols for how to share the data, how to maybe certain standard transformations you do to the data? I think we're interested in learning more about different types of protocols that people have that they'd want to share and contribute within our system. Oh, and just to say, I know before we've talked about too, when you're sampling for OMIC data, oftentimes you're taking a water sample or a soil sample. So that involves getting permits and permission to work in that area too. And so we were thinking about really also developing frameworks where you can share those types of protocols so people can see where others who have worked in certain regions, what work they had to do or they did to get permission or a permit to work there. Oh, Martin, you have your hand up?
It's interesting that you mentioned the whole permissions and so on, because it's Australia and New Zealand is actually quite, I'm assuming it might be similar in Canada and some other areas of the world. The data collection is quite important. So in our best practice guidelines, we put a whole section on that, because we have a lot of First Nations people here. And so Australia and New Zealand, there's a very strong well, there's differences between the countries, but there's a strong drive towards, you need to get permission of the First Nations people as well. And so New Zealand has got quite a strong legal framework on how that should work. Australia is more, there's a legal framework, but it's not as well designed. So it should be part of the protocol as well, for our countries at least. And depending on the kind of DNA you detect, for example, there can be ethical implications for terrestrial DNA, specifically if you do detect human DNA for some traditional owner groups. As in, maybe to explain, in the north of Australia, there's groups where there's a very strict taboo on people who are deceased. So if you detect DNA from ancestors, there's a whole heap of ethical implications that come into play. So it's something in our countries we're really trying to figure out how to work with that. So there's, I don't know if that's relevant for most of you. I mean, I come from Europe. That's not so much a thing, but what we're kind of doing, or what I'm working towards with marine park managers, is I'll have specific collection protocols, because what will happen, there'll be rangers going out to collect samples in biomonitoring projects. So we'll have to decide on one protocol, and that'll be specific, but then we'll have a whole different work for the lab where it goes from there, what the goal is. So we're struggling with the same thing. And I feel if you have a specific goal in mind, like in monitoring a specific thing, it's a lot easier to have at least the collection sorted. But after that, it's how to share it, how to get people to use it is a different question. Because I don't really know for Europe, but Australia, I was actually writing that in the chat. Jodie's work is great, and in IMOS, like a lot of people are aware of it, but the general eDNA Society, there's very little knowledge about the OBPS system, basically in the Omics world. Beyond IMOS, which I'm not sure how to change. Maybe Jodie can come in. I see other people are up. I'll stop talking.
Christina, would you like to share something, and then Anais?
Yeah, just touching up on something that Martin said, because you mentioned that you have rangers going and collecting samples. I was just, wanting to ask you if if you had like questions on the protocol, you specified them, you gave them a protocol and they were supposed to collect samples. How is there any trust issue that they will do exactly as the protocol says? Do they want to modify it to make it easier? Because this might be issues that we may have to track, let's say, somehow in our metadata fields.
Absolutely. So just to be clear, I'm advising managers, so I'm not actually doing those direct interactions. But the big problem in Australia is, I mean, it's just the Great Barrier Reef is larger than the UK, right? So we're talking the scale is Australia is many times larger than Australia. So if we're collecting eDNA just in the marine environment, if we go to the north of the country, it's tropical, it's super hot, there's crocodiles, there's whatever. But where I live in the south, one of our marine parks is sub-Antarctic, so then you've got like Antarctic Ocean. So the range is massive. So depending on who goes out, it's really difficult. And there's one example. There's a researcher up in the north of the country, and she works a lot with indigenous groups that collect estuarine samples for invasive species, and they struggle with exactly what you said. So she had filtering protocols, she had different things, and the reality of the situation is they didn't have time. Plus, it's so stinking hot there that like it would degrade. There's no facilities, there's often no electricity. It's super remote. So she's developed a protocol with just a buffer. So she's just gone collect water, add buffer to it, and bring that to the lab, because that was the reality of the situation. Another researcher in parks in Western Australia, which is essentially half the country, they've got for parks in Western Australia one Omics, one eDNA scientist that will lead all the projects. So you can imagine she struggles, and a lot of the protocols that she was suggested to use, they just don't work, because you go in, it's also terrestrial, you go in an arid region, and she got some, I won't mention which manufacturer, but went, oh, use this, and the first thing that says, oh, don't use when it's over 30 degrees or when there's dust, and she was in the desert where it was 45 degrees. So there's a whole heap of problems that need to be addressed as well.
Thanks. And Ace, would you like to go real quick? You still have your hand up.
Yeah, I think it was funny because it started to say that in Canada we probably have similar challenge, and this is definitely true. That it's not 45 but minus 60 in the Arctic. So basically, we have a lot of indigenous people in the Arctic, and I was working with them to make it simple. So I think really make it easy, sampling kits you can ship by mail. It's so expensive to travel there. So when you can have citizens to work with us, it makes a huge difference, because in my perspective, eDNA is the biggest powerful of eDNA is to survey at large. So for Canada, and Australia, it's they're so big country and so hard to know what happened in the ocean. So if we can make it simple, that's great. But then we have the challenges are that we cannot have ethanol in the Arctic. It's very complex and needs a lot of permits. We need buffer that keep at room temperature. So we're using a lot of Longmire buffer, which is not ideal for the best preservative for when we work in the south and you have lab access. So it's really all of like those limitations help us to decide which protocol basically it's best in which environment, with all of the limiting factors that we're dealing with, and hopefully better understand all of those limitations and how we can compare those results at the end of the day if we are seeing changes. And so here I'm just close to the main versus in the Arctic, which is very far from here, and very different ecosystem. Tides can be very different as well. So the depth, so the sampling protocols, lab are pretty, we are some kind of couple labs in Canada where are doing eDNA like massively. So the communication is quite fluent and we have a lot of decision tree. In terms of sampling design, it's all over the place still, and just which depth which type of filters, which buffer, all of that type of things is very hard to know which one to decide to take. And we're trying our best, but so far communication is still hard to know everything that needs to be considered.
Thanks. I think, one thing we've talked about is trying to talk about or label why you made certain changes, or if you made a change because that's the environmental limitation you had, or if the change, was driven by something else. Raisa, would you like to add something?
Yeah, I was just, thanks Anais for that comment. That reminded me of the sometimes having restrictions in the country that only allow you to use certain chemicals or certain equipment or anything like that. So actually, while developing the template for the protocols, that was one of the steps that we thought about. And the way that we thought to approach it was to have a very clear resources list. So this is exactly what you need, or exactly the chemical that was used, to not only have that as part of the text, but very openly at the front of the protocol, so that you can already check, okay, this includes ethanol, so we can't really use that. Let's move on to another one. And then hopefully also have machines support the search through all of that. But yeah, it's been really interesting to learn about some of the implications depending on where you are with the DNA from deceased people from Martin, or with the ethanol from you, Anais. So exceptional how we're all working in such different conditions and trying to make it work.
Frank, would you like to share something?
Yeah, I think several people have mentioned OBON and the projects that people submit to the Ocean Decade for endorsement, and then they can be affiliated to programs like OBON. Yet there's a whole family of other groups, like we've heard here. There's some under the specific Marine Biodiversity Observation Network that are now starting a new cycle of 5 years of funding, and they all are doing eDNA now. And many of them, I think all, except maybe yours, are not affiliated with OBON. So how do we get this community together without having to go through the, I would say, the protocol of writing an Ocean Decade project proposal, rather really just jumping right in and making sure that we do get that community together that has this discussion? As you know, you have the meeting every 2 weeks. We have an MBON meeting on eDNA once a month. They're overlapping because some of you participate in both, and yet I wonder, how do we make this community tick?
I think for me, I'd like to think about what benefits we can provide. But yeah, would someone else like to jump in? I thought I heard someone. But I guess I'll just say real quickly, like, I know in our lab it's a challenge, like I was mentioning before, linking the exact protocols that were used with the exact, data sets and keeping track of all the changes we've had through time. And so I think, for me, a lot of the carrots that we're providing is a system that can keep track of that for your lab, and then maybe also a system that can make visible what current protocols are in use by other efforts, and then help people find out what other types of protocols have been used nearby you. So I think the more we can kind of develop functionality that helps people improve starting up their own OMIC observatory network, or managing their protocols for their current one. And the more we can move towards then being able to apply that to how we can, combine these data sets together, I find that very motivating. So yeah, I guess it was worth, having a discussion now about, what would be, you think, the most motivating ways to make or to encourage participation in this network. I know I heard, somebody else has suggested, what if we try to go in and have like journal requirements, or, the carrot versus the stick kind of argument. But I think right now I like to think about carrots of what we can provide by getting people to participate in this kind of framework.
I might quickly jump in there. I was just on a workshop a few weeks ago of an Australian government initiative to standardize all kinds of ocean best practices, essentially, which Rachel Przlowski was leading. So, and we had a discussion about how do you get people to buy in, and even those that are very well designed, it's not a legal requirement. Like, it's very highly advised by funders, please use these protocols, but they can't, as I understand it, at least, because it's not my field, they can't officially force people to use them. And so I think it depends on, we had the same developing the best practice guidelines. How do we kind of formulate this that there's an incentive for uptake without going restrictive? So we kind of made the distinction, if you do research, do whatever you want, right? You can figure out what goes on. But then I hope Jodie will comment, because I feel like this is very much her field. Like, if you've got well-organized monitoring programs like IMOS, you can standardize. And I know from the people I talk to for my day-to-day job, there is quite a demand on how do we do this properly, partially because obviously they want it done right and they want to use best practice, but also very often they're not experts, like going back to whoever collects it. So I guess it depends on the situation. And yeah, I'm looking forward to having Jodie's idea, because she just mentioned that she'll talk about OBPS and how to motivate the community to use them. Yeah, funding, like, I don't know if funding agencies would say, please use this, that might be useful. But yeah, anyone's opinion would be interesting here.
Thanks, Martin. I do agree that funding quite often plays a very, very big role. I mean, not everyone can afford I think it's about $15 Australian now per Sterivex filter within their projects. You know, we use Sterivex filters because they're encapsulated. A lot of the people that collect our samples aren't microbiome experts that are, you know, they're going out all over Australia. So we find that is the safest way to avoid contamination, but they're expensive, especially if you've got a big project with big spatial scales, big temporal scales. So, you know, funding is a very big part of it, and we're fortunate that IMOS provides that funding to us. So all of the consumables come through CSIRO out to the other institutions that are collecting for us around Australia, and that's everything from, we provided the pumps to the tubing for the pumps, you know, the Sterivexes, then all the samples come back to us. We do all of the extraction and that gets sent to a sequencing facility. So yes, that is standardized, but it takes funding. So yeah, in my experience, that's been a really big part.
Can I jump in and say something here? To ask the question of why are we doing this? Why are we wanting to standardize? In the Southern Ocean, it's to have comparable data sets, and in the global ocean,
Oh, you muted yourself.
well, I'm not the expert, so probably I'm better on mute, but it's the big what for. What are we doing this for? And do we want to have globally compatible data sets so we can put it all into a model and manage the Earth system? Is that the what for? Is that why we're doing this? Or, you know, I know people talk about stakeholder needs and they have to deliver. Yeah, so I'll go back on mute.
No, I think absolutely that's a huge end product that we want. And I think the first step is understanding what protocols were used to generate which data sets. And then we can start thinking about which data sets are comparable and at what level to each other, and how better we can make them more comparable. Inés, would you like to?
Yes, I think the answer was good, very good. It's actually comparable data, but there's also a lot of people that are using a method and they don't necessarily know why. It's basically they use it because it has worked once, so they keep it. However, there's so many, like, we learned so much in the last 3, 4, 5 years. Like the number of papers has been just massive. So now we need some kind of making sure that there are some limitations, of course, but there are also stuff that can just being done and that are cheap, that are easy. So we need to basically advise all of those companies that are developing right now, students that are starting different labs. Like, I mean, it's just so many different, they're not just experts that are interested about eDNA. There are also a lot of users. Managers are receiving data and they don't know how if they can trust the data or not. So having some guidance on which method is best, I think it's even if we're not talking about the perfect standardization, I think that makes a lot of sense with everything we have running. So compiling all of that knowledge. So in my perspective, it's very timely. Like, it's perfect now because we have learned so much. So now, even if it's not perfect, but I think we are ready now to have some guidance at least, and hoping to have this perfect. And how does that change really all of those steps, the final interpretation of the data.
I think just to add, like, now is a good time to try to start, like, for example, the process that BBOP wants to do of trying to compare protocols that are in use. And I guess also thinking about, there are a lot of groups that are trying to work towards protocol harmonization or that kind of thing. And we want to make sure we're not reinventing the wheel of anyone who's already working in this sphere, and see like where we can work together to kind of accomplish this goal, instead of everyone kind of splitting off in their own separate networks and developing similar systems. So I think that's a big goal for us too, that we want to try to connect with others and let them know about the work we're doing and see where we can work together.
I think you highlighted a good point in terms of people, if they have not been successful, they will not publish it. So basically, we'll never know. So you reinvent the wheel all the time. And 10 years ago, I was not successful for a couple of months. And this was very frustrating as a postdoc. You need to publish quickly and things like that. So then it works, and then you stick with that, whereas you could improve it. So
right.
So
some guidance
about sharing best practices are definitely important.
Right. I know one thing we've brought up before earlier is also the sharing of like failed practices, or I know in our lab, we've tried protocols that then didn't work. So I think one thing I'd like to, we haven't discussed it really in terms of BBOP yet, but I think that would also be an interesting application where we could show, I'd be happy to show, our protocols that didn't work for us in our environment, and how that what changes we made maybe that led to like our current protocols now. But I think, we're running a little short on time. We're running up on, I think about 10 minutes left. We've covered a lot of what I wanted to talk about today, but maybe except kind of the question of, does anyone see any gaps or something that's missing in the BBOP framework that we discussed today, or anything that, we're not seeing because we've been working on it for a while, or like, I guess we just also wanted to get feedback from others working in different parts of the world or different systems. If you think there's anything that we're missing. Jay, would you like to say something?
I haven't heard address capacity building or capacity sharing. Let me use the latter term in the discussion today. Is that part of the efforts you have?
I think it's a big motivator to make protocols discoverable. I guess when you think about building capacity, it's also about for people to discover what protocols will work for them or could work for them in their environment. And so I guess that's kind of how I've been thinking about it. I don't know if others want to jump in too, but I think, when we first met in 2020, we talked about wanting to have an ability to sort through all these different omics protocols or be able to tell which ones could be applied in which regions. And so that's kind of what led into our developing this MIOP, these minimum metadata terms about the protocols, and then also this kind of framework, really making clear exactly what people are doing in different areas. So to me, that links into capacity building, but I don't know if Christina or Reza or others would like to jump in with how they think about it.
Maybe briefly, I completely agree with what Katie said. And then on the actually writing the protocols using the format that we're developing, we're also have in the pipeline or in the plans to prepare some video tutorials and guides on how to best work with the templates in case anyone's not familiar with Markdown, how to work with that for the first time. I know that's only a very small part of the capacity building, but it's still essential to make sure that with what we're doing in trying to bring people together to share their protocols, we're not excluding anyone, just because of how we as a group thought the best way to approach that.
Oh, Sarah, would you like to jump in?
Yeah, I didn't introduce myself. Sorry, I'm Sarah Stinson. I work in California for the State Department of Water Resources. Just graduated from UC Davis in their ecology graduate program. And one of the challenges I'm facing in my new job is that I'm setting up a brand new genetic monitoring laboratory for the state. And in this department, this is the first type of laboratory for us. And so we're navigating a system that doesn't know what to do with us. So things like data storage, just some amount of justification of the expense of doing molecular work has been required, although I think most people are very excited to have us on board with what we're doing. And one of the groups that I've been involved with, which is helping us to work through this startup initiation process, is the California Molecular Methods Work Group. And they've been really helpful with bringing together different groups from mostly focused in California, but different agencies, academics, nonprofits, different areas, indigenous tribal groups, and trying to standardize our protocols within the state, because a lot of us work in really challenging systems, like in estuarine systems where we have a lot of turbidity, or water temperatures in the summer months can be pretty warm. So I think that echoes some other comments from earlier, but we're facing all these small and large hurdles of, okay, well, now we have the MySeq. What are we going to do with all this data that we're going to generate with it, and things like that. So I don't know if anyone else has that issue, or if that's not directly relevant to other people in the group, but it's been an interesting challenge in my new position. So I'm glad to be here, and it's been great to listen to everyone's opinions.
Thanks. Yeah, that just reminds you of, how many people are working in our sphere kind of in California, or how there's these different regions that are highly observed. And how at least, maybe that's a motivation in a regional capacity to try to get everyone to talk together and compare our data sets. And I've gone to some of the molecular method working group meetings and thought they're wonderful. So it's a really great network to be in.
Yeah, there's a lot of very knowledgeable individuals in that group, and Susanna Thoreau is the leader of that. And I kind of thought maybe other members would be present, but I think maybe I'm the only one. But she's been great at focusing our conversations and talk about talking about consensus building from different backgrounds, different fields, etc.
She's been part of our work too. So she may be able to come to some of the other meetings this week. But yeah, she's great. Well, I guess we're kind of wrapping up. Does anyone else want to share anything before we close? I guess just to briefly go over, so tomorrow we'll be meeting at the same time. And so tomorrow we really wanted the focus to be talking about our minimum metadata that we've developed for protocols. And so there it's kind of discussing, you know, the set of data that we've thought is necessary to describe different protocols and to be able to make them more findable. But we'd like to talk about what others think too, and get feedback. You know, this set of metadata we may try to link in with the GSC, the Genomic Standard Consortium, as being something that labels protocols. But that'll be discussion for tomorrow.
Katie, I missed it, but does the protocol, the methods protocol, right now link or include applications of some sort? So I think going back to the decision tree, if I want to do this, I use this method, or if I want to do this other thing, it goes that way.
So we have a purpose category, but yeah, so we have a purpose and we have, you know, the environmental context of like what's your target, what environmental medium are you sampling. So we have some terms that kind of try to get at that. But, you know, just from talking today with all the, you know, unique temperature requirements or other things, it makes me think if, you know, temperature is also important about to know what temperature protocol has been applied in. But I think part of our discussion tomorrow is we want to really think about how we pare things down to a minimum. And then maybe after we have that minimum, we can then build like certain packages the same way the GSC does about different types of sampling or different, you know, what are the metadata requirements for protocols describing, you know, in this network or this kind of region. So I think tomorrow we want to focus on getting to a minimum, but then also talking about, okay, now that we have this minimum, what are other things that are nice to have depending on what you're doing or where you're working or that kind of thing. So that'll be part of the discussion. Oh, George, yes, sorry, I didn't see your hand.
Howdy, everybody. This has been really enjoyable. And I'm just going to plant a seed in the chat for Jay and Frank and you who are really plugged in at the highest level. It's related to, can ocean best practices perhaps help UNESCO's endorsement and the billionaire foundations accelerate doing what they're trying to do to get the, so that's the seed that I'm going to put in there. I've got 4 or 5 things to consider, and maybe we can talk about it at the end of the session tomorrow or something like that. Just again, it's an idea I've pitched to several other people. I think it has some merit, but I'll put the ideas in there in the chat, and then we'll see if anybody else thinks there's a possibility that the committee could do something to accelerate and amplify the, you know, there's obviously a big disconnect between hundreds of $trillions pledged for nature for the decade and the amount of money that we can hear and read and validate that's actually been given to anyone. So how do we fix that? There's the desire from the money. There's obviously the need we've all been working on for 30 or 40 years. How do we accelerate? The good people with money want to help. Let's help them help us if we can. So I'm sorry to rant, but that's just, it's just really frustrating after 4 or 5 and watching 2 years after somebody gets endorsed and $1 hasn't happened. It's just mind numbing. So anyway, sorry to rant, but that's where I am right now. Kind of. Anyway, I'll get it and paste it in. I've got it in a document here. I'll put it in there and I'll shut up, quiet, popcorn, back to you guys.
Thanks, George. Yeah, I think the money question's a good one because, you know, a lot of us are volunteering our effort to try to build these kind of systems. So getting support would be great. Yeah, well, I think we're at the end of the discussion period now, though. And thanks everyone so much for coming today. And I hope some of you can make it tomorrow or throughout the week too. We have one more session tomorrow and then one more session on Wednesday, same time. But thanks, everyone.
Thanks, everyone. Thanks,
Thank
Kate.
you.
Thank you very much. Thanks.
Bye.
Bye.